Did all of you guys finish the practical? Okay, I just wanted to show you quickly. I know you finished the practical. I don't know if any of you caught this, but I posted some practical study materials. So there's this new drive. So for the next one, just so you know, it's going to be here and I'll put my practical study materials. So I have this has like a bunch of the histology labeled. I mean, it's not the best, but like you'll have just so like the histology is in one place for you to review before you take it. And then this document is super long, but I would take notes on all of the labs before. So that's just my notes on all the labs. So I'll post that for the next practical too. And did everyone sign in? Thank you guys. Okay, so for this week, I thought we were going to be on respiration, but we're not yet, right? Okay, so I already have those slides made, so I'll probably post them after this so you guys can start looking at them if you want, if you're ahead. But we're going to do immune part 2, so we're going to get a little bit further into specific immunity. Office hours are tomorrow, email, and then practical study guides and other study guides are on Husky CT. Okay, so here's just a quick review from last week. There are 2 divisions of the immune system overall. You got non-specific and specific. So just to review, non-specific, present from birth, does not respond to a specific antigen, and it involves external and internal defense. So then the skin, mucous membranes, proteolytic enzymes, acids, lysozyme, interferons, and the complement system. So again, these all respond to any pathogen, nothing specific. Specific immunity needs to be learned through exposure, so you can produce antibodies for a specific pathogen. And this is where we got the T cells and the B cells. And these have specific receptors that match up with specific antigens to initiate a specific response. So then you have 2 divisions within specific immunity, cell-mediated, which is your T cells, and the humoral response, which is your B cells. And we're going to look closely at those today. So I color-coded the slides. So orange slides are going to be cell-mediated with T cells, and then the blue slides will be the humoral response and B cells. And then I took this from the lecture. It just kind of breaks down again. We're talking about specific immunity, T cells in the cellular response, and B cells in the humoral response. So this is also from last week. There are 4 types of T cells. Just to remind you, there's the cytotoxic T cells, helper T cells, suppressor T cells, and memory T cells. The cytotoxic T cells respond to an antigen displayed on MHC1, whereas the helper T cells recognize antigens on MHC2. So helper T cells also help activate cytotoxic T cells, and they promote the humoral response. They also help activate B cells. Suppressor T cells kind of just turn off the cellular response by releasing inhibitory cytokines, because we don't want to have an immune response all the time, so they just help turn it off. And then memory T cells go through clonal expansion, so they just replicate, and then these cells are just in your body and ready for the next time that the same pathogen comes in, and they can initiate the response quickly. So this stuff we did not talk about last week. these are some features of T cells. So T cells have both a T cell receptor, or a TCR, and a cluster differential marker, which is known as a CD marker. So the T cell receptors recognize a specific antigen that's being displayed by the MHC and binds to the antigen. And that's the main point for TCRs. CD markers, these are membrane proteins on the T cells that help activate or bind the MHC, but they don't bind the antigen directly. The CD3 marker is on all T cells, and the CD28 is on the cytotoxic T cells and the helper T cells receptor. Yeah. And then there's also CD8 and CD4. And CD8 is on the receptor of cytotoxic T cells, and CD4 is on the receptor of helper T cells. And those just help the T cell bind to MHC. So they help the cytotoxic T cell associate with MHC1, and they help the helper T cell associate with MHC2. So they help them dock. And I have a better visual of that soon, so it's more clear. But here's VDJ recombination, which is variable diversity joining recombination. And this is the maturing that the T cell receptor goes through to allow it to recognize different antigens. So the DNA kind of just scrambles when it's being matured in the thymus, and that just allows it to recognize different pathogens. So it gives the immune system more kind of like of a magnitude of diversity of things that it can respond to. Yeah. Here's some, this is T cell activation. So we start with a virus that enters the body, then an MHC is going to then display that on the plasma membrane. So the body recognizes this virus and displays a little portion of it on the cell membrane. So then a cytotoxic T cell that has the receptor for that antigen can bind. And then CD8 helps it bind to MHC. An important factor here is co-stimulation. So CD28 on the cytotoxic T cell binds at CD80 or CD86 on the cell that's displaying the MHC1, and then CD8 on the T cell also binds to the receptor of MHC1. So I have another slide that makes that more clear, but it's important that those two factors happen for the activation of the T cell. So you can see in the picture in the corner, co-stimulation is referring to the two dark blue receptors binding and the black CD8 binding to the MHC, if that makes it more clear. And then we go through clonal expansion to activate more T cells, and then the activated T cells secrete perforins, which kill the body cell by poking holes in it. They stimulate apoptosis or cell death, and they secrete lymphotoxin, which disrupts cell metabolism. And then we're also going to produce some memory T cells, so then when the pathogen enters the body, they're already suited up and ready to attack that specific antigen. Okay. So there are 3 binding sites that the MHC cell binds to on the T cell to activate it. So the first one is at CD8. So CD8 is one of those markers on the T cell that binds to MHC1. Next, you have CD28 on the T cell, which binds to CD80 or 86 on MHC1. And a combination of those two binding sites is co-stimulation. And then there's a third binding site, which is just the antigen in the MHC binding to the actual T cell receptor. Does anyone have questions on that? Because I know it's like really confusing with all the numbers and okay, I'll move on. Then we have helper T cell activation. So this begins with an APC or an antigen presenting cell that finds a pathogen and then displays a piece of the pathogen on its MHC2. So then an inactive helper T cell with a specific receptor for that antigen presented on MHC2 binds. And here, instead of CD8, we have CD4 that helps it dock to MHC2. So if I go back, instead of CD8, it's CD4 that's doing the same thing, just helping the helper T cell bind to the MHC2. And then CD28 on the helper T cell binds to CD80 or CD86 on the antigen presenting cell, which is the cell with MHC2 on it. And then again, the third binding site is when the antigen directly binds to the receptor. And then again, we're going to have clonal expansion of the helper T cells, and then some send out cytokines, and the cytokines stimulate both the cytotoxic T cells and B cells, so it stimulates both the cellular and humoral response. And the cytokines also stimulate macrophages. And then again, some memory T cell, helper T cells, so they're ready if the pathogen is reintroduced into the body. I took this from the lecture slides again. So this just kind of shows how the antigen presenting cell is kind of like any nucleated body cell. So any body cell that then presents the antigen on the MHC. So I like that visual just because it's kind of hard to wrap your mind around. So it's taking in a little piece of the pathogen and just holding it out. And then the T cell receptor for that specific antigen can find it and bind. And then the CD markers that we talked about help it bind specifically. And then after it's activated and all bound, you got activation, clonal expansion. If it's a helper T cell, you release cytokines. Yeah. So this is just co-stimulation that we talked about written out. So it's those two factors where it's binding of CD8 if it's a cytotoxic T cell, or CD4 if it's a helper T cell, to the prospective MHC on the cell that is presenting the antigen. And then also binding of CD28 to the presenting cells CD80 or 86. And if you have a lack of CD28 specifically, then you have a weakened partial immune response, and the T cell could become anergic, which means that it's not responding to a specific antigen because it's lacking that marker, which is preventing it from binding and initiating a complete response. Do you guys feel good about the CD markers and what they bind to? Or do you want me to go over it again? Or yeah. Okay, I'm going to go back to this. So, first, CD8 is on the cytotoxic T cell. If it's a helper T cell, it'd be CD4. CD8 or CD4 binds directly to MHC. CD8 in a cytotoxic T cell goes to MHC1. CD4 would go to MHC2. So that's just helping the T cell kind of stick to the MHC, which is presenting the antigen. Second place where we're binding is CD28. CD28 is in both helper T cells and cytotoxic T cells. And it's just on the membrane. And then we're binding to CD80 or 86 on the antigen presenting cell, which is just the cell that's infected with the pathogen that's presenting the antigen. And then last spot is between the actual antigen and the T cell receptor that's binding to the antigen. Hopefully that's more clear. It's a little confusing, but hopefully the visual helps too. Here's another one that shows a similar thing, but it looks a little bit more confusing. So I think I'm going to skip over that because there are more numbers that I don't think we need. Okay. So now we're moving over to the humoral response in B cells. And this is just a reminder of the functions of the humoral response. So it's activated by helper T cells releasing cytokines that we just talked about. And then, as you guys talked about in lecture, it also can be activated by B cells themselves, which is direct stimulation, which we go over later. And the B cells have antibodies on their cell surface. And antibodies serve as specific B cell receptors that recognize a particular antigen. So they're analogous to the T cell receptors. These are what are recognizing the antigen in MHC. Or no, they're just recognizing an antigen. And then once they're activated, they release antibodies if they're mature plasma cells. If they go to maturity. So like I said before, they can be activated directly by B cells, or they can be activated by cytokines and the helper T cells. So direct stimulation is when a B cell engulfs an antigen using its membrane-bound antibody and then presents it on its MHC2. And then direct B cell stimulation is that limited response from the B cell that causes it to produce more antibodies. And then they could also be activated by helper T cells. So the T cell receptor of the helper T cell binds to the B cell, and then the B cell is activated, releases cytokines that help to activate the B cell, and then the B cell goes through clonal expansion, makes more activated B cells, and then those can mature into plasma cells that can release antibodies and become full of rough endoplasmic reticulum, and that is what helps make the antibodies. So important things to note about B cell activation is that there's no co-stimulation. We don't have the CD8, CD28 markers binding, and that it also makes memory B cells, like both cytotoxic and helper T cells. Here's a visual that I took from lecture, so you could see the B cell activation and then the production of antibodies from the plasma cells. And then this is from last week too. So then after the antibodies are produced by the plasma cells, they go off into the body to do a bunch of things to try to protect it against the pathogen. So one is that they directly neutralize the pathogen by coating it with a bunch of antibodies. So blocking the pathogen active sites kind of deactivates it. Another function is agglutination, which is, so they kind of clump together since all of the antibodies have two binding sites. So they prevent the pathogen from kind of moving. They can activate the complement system, attract phagocytes, and activate and call natural killer cells. And then these two next slides go over the antibody structure. So there's both a heavy chain and a light chain, and both of them are made from immunoglobulins in the plasma. And so there are two heavy chains that come together to form kind of the stem of the antibody. So this is like the stem or the base. And the Fc segment determines class, so it varies based on where the B cell is stimulated. And then there are 5 classes, and it looks like the first one got erased. But I missed that one. But then there's IgE, IgD, IgM, and IgA. And yeah, so then those are just different classes of the stem based on where the antibody is produced. And then the light chain are the variable regions. So these are the two branches of the antibody, and this determines the specificity of each B cell to bind to a pathogen. So it's specific for a specific pathogen or antigen, and it recognizes the antigen and binds to it. And B cells undergo a scrambling process that gives each variable segment a unique epitope region. So here's a visual of an antibody. You can see the heavy chains on the bottom and the light chains on the top. The heavy chains kind of go throughout the whole thing, but then the light chains are only on the top. You don't really have to know any of that. It's just so you can see. But the antigen will bind at the top, the antigen binding sites from the, And then there are 2 types of antibodies. Free antibodies are circulating through your fluid to try to bind an antigen, and bound antibodies act as those receptors on the B cells. And the variable region is what's binding the antigen, and the Fc region is what's bound to the plasma membrane. So it kind of extends from the B cell like a Y. And then antibody generation occurs during development in bone marrow. Just wanted to add that in there. Another important feature of B cells is that they can undergo class switching. So this is just when a B cell changes the types of antibody that it produces. So this is due to a rearrangement of DNA that changes the way the protein is expressed, and the constant region that we talked about, the heavy chain, switches classes. But the variable region, which is the top of the antibody, stays the same. So the recognition or the type of antigen that it is recognizing doesn't change, it's just changing class. And that just changes antibody distribution in the body. It doesn't necessarily change like recognition of the type of antigen that it's recognizing. And then there's a primary response and a secondary response of B cells. The primary response just refers to the first time the body is exposed to a particular pathogen. It takes about a week to produce antibodies. IgMs are the first produced. And then the secondary response is possible because of those memory B cells. So it just takes less time to initiate a response. And it prevents more of a severe illness on the second exposure. And then I just wanted to add this in here because I think it's interesting to consider the hygiene hypothesis. This just proposes that our immune system, like if you're not exposed to a lot of pathogens, it doesn't learn to recognize self from non-self, and it starts to attack itself, causing an autoimmune disease. So this is like a theory that if you grow up in an environment that you're not exposed to a lot of pathogens, that the body like doesn't know how to use its immune system. So it tries to explain why people that have contracted less infectious diseases tend to have higher rates of autoimmune diseases. But it's really more about like microbial populations in different communities in the environments that people live in. Okay, let's move to the top hat questions. Okay, which of the following players are players in non-specific immunity? So you can select as many as you want. Oh, does it? Mm-hmm. Let's edit. Oh. Oh wait, I think I know how I can do it. Let's see if that changes it. Okay. I'm gonna go through because I have other ones that I want to accept multiple. Okay, I think that's good. Okay, let's see. So the question asks about non-specific immunity, which is the unlearned response to any pathogen that enters the body or the body's exposed to. So you guys did pretty good. The complement system, just remember that's a part of non-specific immunity. It's an internal non-specific response that helps, produces like proteins to help the immune system attack. So then B cells, any type of T cell antibodies are both part of specific immunity. I'm gonna go off so we can't see what people say. Oh wait, I'll read the question first. TS cells are able to terminate the cellular response by, and those are the suppressor T cells. Oh, nice. Okay. Yeah, so by releasing inhibitory cytokines, they can end the T cell response. What three regions do cytotoxic T cells bind to on the antigen presenting cell, which displays MHC1? Yeah, that's pretty good. So the three sites are in the cytotoxic T cell, CD8 to MHC1, CD28 to CD80 on the antigen presenting cell, and then the antigen to the receptor. I saw some people picking antigen to MHC1. So there is an antigen in MHC1 because MHC1 is presenting that antigen to the outside of the cell, but that antigen binding to the MHC isn't helping the T cell bind. It's kind of just there. So it's not a binding site between the T cell and the antigen presenting cell. Co-stimulation of T cells involves the binding of two separate pieces of cell machinery. What are they? I think that's supposed to say CD8/CD4. So it also, I was also, I think, yeah, wanting you to pick the first one, but the second one is good too. So it's both binding of CD8 or CD4 to the antigen presenting cell. Bless you. And then the CD8 to CD80 or 86. So I should edit that. A lack of CD28 may make cells anergic, which means, I'm going to fix this. So anergic means that the body will not be able to respond to a specific antigen because a lack of CD28 on a specific T cell may prevent it from recognizing a specific antigen on a pathogen. B cells exhibit co-stimulation. True or false? So this one is false. Co-stimulation, again, refers to the CD4, CD8, CD28 binding, and B cells don't have that. They engulf the antigen.
Question.
Yeah.
Do B cells produce MHC2?
Yeah.
So like, wouldn't they still like if something like the T cell is going on to the B cell, wouldn't it be co-stimulation like both of them together?
Technically, yes. If a B cell is acting as an antigen presenting cell.
Because like, okay.
Yeah.
Because like, don't all B cells like have the MHC2?
I think it's referring to B cell activation. When a B cell is activated, it does not, when it's self-activating, it does not need co-stimulation.
So when it's like being activated by a helper T cell, then it does need co-stimulation? Because a helper T cells like release cytokines? Or is that like, cytokines is what's stimulating it?
If a B cell is like holding up an antigen on its MHC, it could possibly use the same mechanisms that we were talking about before, but I don't think that will come up specifically. Like it would have to specifically say like a T cell is recognizing an antigen on an MHC on a B cell. That's the case that you're talking about. So I don't think that will come up. Like in general, when they're activated, they don't use co-stimulation, but in that case, maybe they could. I'm not entirely
That's
sure.
fine. I just didn't know from the lectures and stuff because I thought MHC2 is only on B cell, dendritic cell, or does it one more? But those ones. So just
I can look.
Okay,
I'll look more into it after too. Yeah, to double check. B cells can be stimulated by. That was a good question though. Okay, just to answer your question,
Mhm.
you are right. The MHC2s are displayed by professional antigen presenting cells, which is like the B cells, macrophages,
Macrophages, okay.
whatever. But I think when they say co-stimulation, it's referring to like the activation of that cell. So
Okay.
B cells don't need co-stimulation to be activated,
Mhm.
but they use co-stimulation technically when they're activating T cells.
Okay.
That
Okay, that makes
makes
sense. No,
sense,
I was
right?
just like super confused with that.
No, because that's a valid point. Okay, so now for this one, yeah, B cells can be stimulated by B cells or helper T cells. What is it called when a B cell changes the antibodies it produces? Yes, so this is class switching when the antibody kind of like alters the DNA or what DNA is being turned on in the heavy chain. If you're confused with VDJ, this has to do with T cells, the T cell receptor kind of changing up that region to recognize different antigens. But you guys got it. And then last one is one from last week. To review, what are the 3 secondary organs of the lymphatic system? So just to remember, the primary organs, I believe, are the thymus and red bone marrow. Secondary organs, nodes, nodules, and spleen, just to keep those straight. And that's all I have. We can end a little early if you guys have to go. Feel free to stay for any questions. And then I also linked my notes if you haven't been using them, if you want to check them out. But thanks for coming.
Just quick, will the respiratory stuff be on the quiz this week?
1 to 3. Lectures 1 to 3.
Okay.
Oh wow. Okay. Thanks.
Nicole just told me like at office.